how did life on Earth actually begin? I mean, it's one of the biggest, most fundamental questions in all of science, right? And for the longest time, we've pictured the answer lying somewhere in a dramatic primordial soup. So we've been looking for clues in scenes just like this one, a violent, chaotic, early Earth. But what if the first, most important clue, the key piece of evidence, isn't 4 billion years in the past? What if it's all around you, right now, in the form of, well, slime? You've definitely seen this stuff. It's everywhere, on rocks in a stream, inside pipes, maybe even in the corner of your sink. It's called a biofilm, but here's the thing. This is not just random pond scum. It's an incredibly sophisticated and coordinated community. In fact, something like 80% of all prokaryotic cells live this way. It's one of life's most successful strategies for survival, hands down. And to see why, just look at the difference. A free-living microbe? It's out there all on its own. It's vulnerable, it's isolated, and it's burning a ton of energy just to stay alive. But inside a biofilm, life is communal. The whole group is protected by this shared matrix. They can share nutrients, and they save energy by working together. Going it alone is tough, but life in a biofilm, that gives you some incredible advantages. Okay, so now let's rewind. Let's go back to the original scene of the crime, that early, hostile Earth. Because when you really look at the conditions back then, the beginning of life starts to seem less like a miracle and more like a total impossibility. I mean, the challenges were just immense. You had no ozone layer, so deadly UVC radiation was just baking the surface. Temperatures would swing like crazy. Any interesting molecules that did manage to form were probably way too spread out in the huge oceans to ever meet each other. That's what we call the dilution problem. And even if they did meet, water itself was the enemy, constantly trying to break complex molecules apart. So that's the central puzzle, right? In a world this hostile, how could fragile, complex chemistry ever get organized and stay protected long enough to become biology? It's a massive question that has stumped origin of life theories for decades. But a new idea suggests that maybe we've been looking in the wrong place this whole time. What if life didn't start out in the chaotic open water? What if it was nurtured inside a protective, stable cradle, a sort of prebiotic slime? And this is where the prebiotic gel comes in. The whole idea is that simple compounds that were likely all over early Earth, things like clays, silica, simple polymers, could have formed these soft, squishy matrices. You can think of it like a primitive, non-living version of a biofilm, a safe neighborhood for chemistry to finally get its act together. So what makes a blob of gels so special? Well, according to this hypothesis, these gels weren't just sitting there. No, they were active chemical incubators that had some pretty serious, well, superpowers. And this gives us a fantastic overview of what these gels could do. You can see it acting as a container, a shield, a reaction booster, and even a little engine. This one structure solves a bunch of problems all at once. So let's break down the 4 key superpowers that might have made these gels the perfect incubator for life. First up, concentration. The gel's web-like structure basically acts like a sieve. It traps all the important molecules and stops them from just floating away into the ocean. This totally solves the dilution problem, right? It forces them all together, which dramatically increases the chances that they'll react and build more complex stuff. Superpower number 2, protection. These gels were a powerful shield. And get this, experiments show that certain gels, especially ones made with common clays, could block over 90% of that deadly UVC radiation. That creates a safe little haven inside for all that fragile chemistry to happen. Third, they were reaction boosters. Now, a huge problem for early chemistry is that water loves to break apart the long molecular chains that are essential for life. But inside a gel, a lot of that water is, well, it's busy. It's bound to the gel itself. This lowers the amount of free water, which tips the chemical balance in favor of building those long chains, not breaking them apart. And finally, the 4th superpower, energy. Gels could trap minerals and other molecules, creating a system that could actually pull chemical energy, like electrons from pyrite, and use that energy to power the creation of even more complex molecules. It's like a primitive power plant, a really critical step towards a self-sustaining metabolism. So this is the crucial question. If these gels were so perfect, so amazing, why did life ever leave? Why aren't we all just really sophisticated slime today? You know, the gel was the cradle, but at some point, life had to move out of the house. The theory suggests a step-by-step evolution, a kind of path to freedom. It probably all started inside the safety of the gel, where new structures, like little compartments or primitive vesicles, could form. Over time, these compartments could get more and more complex, becoming the first true protocells, all while still being nestled inside that protective gel. But eventually, they'd become sophisticated enough to break free. And this process could have ultimately given rise to LUCA, the last universal common ancestor. This is the single-celled organism that sits at the very base of the evolutionary tree. It's the ancient great-great-great-grandparent of every plant, animal, fungus, and bacterium you have ever seen. The gel was the nursery, but LUCA was the first graduate. Now, this whole gel-first theory doesn't just rewrite our own origin story. It completely changes how we should be searching for life somewhere else, out there in the universe. See, for decades, our search has been guided by this idea of life as we know it. We look for cells, for Earth-like biochemistry, but as the source material points out, that's a huge limitation. What if life on other worlds never made that jump to free-living cells? What if it just stayed in the gel? This opens up the absolutely wild possibility of xenofilms. I mean, just imagine it. Alien biofilm-like structures, cosmic slimes built from a totally different set of chemical building blocks. They might not have DNA or proteins or anything we recognize, but they could still be complex, evolving, living systems. We need to start thinking about how to find life as we don't know it. And that leaves us with a really fascinating final thought. We spend so much time imagining what alien creatures might look like, right? But maybe we're thinking about it all wrong. Maybe the most common form of life in the universe isn't walking around on two legs, but is a complex, evolving, and maybe even intelligent slime, just thriving in its own little cosmic cradle.